We, the final year students of ACAR College of Agriculture and Research, are undergoing the project work on the title Characterization of Molecular Traits of Traditional Land Races and Isolation of Genomic DNA. In this video, we'll see about the isolation of genomic DNA of traditional land races using CTAF buffer method and gel documentation. Now let us see about the genomic isolation of DNA in brief.
First step, collection of plant sample using the mini cooler. Now we are taking the mini cooler from the deep freezer and moving it to the field for plant sample collection. Now the plants are collected with the help of scissors and Ziploc cover. We are taking the plant leaf tip placed in the Ziploc cover. Finally, the Ziploc covers are placed in the mini cooler and brought to the laboratory for further step. Now we are preparing CTAP extraction buffer. For that, we are taking 90 ml of beta mercapita ethanol using the micropipette and poured into the beaker. Now we are taking CTAP extraction buffer 9 ml in the measuring cylinder. And poured into the beaker containing beta mercapita ethanol. Finally, we are taking CTAP powder using the glass rod These chemicals are mixed thoroughly and kept in the water bath for 5 minutes. Then we are taking the plant sample and weighing 0.5 gram. Then the plant samples are transferred into the mortar and pestle for grinding it. Now we are adding 600 ml of CTAP extraction buffer using the micropipette and grinding the sample nicely. Then we are transferring the sample in the F&F tube and preheating it for 65 degrees Celsius. Preparing chloroform and isopropyl alcohol solution. For that, we are taking 24 ml of chloroform in the measuring cylinder and 1 ml of isopropyl alcohol. These two are mixed in the ratio of 24:1. Then we are adding 600 ml of this isopropyl alcohol and chloroform combination into the F&F tube. Then we are placing under mini centrifuger for 5000 rpm for 15 minutes. After 15 minutes, we are removing the F&F tube from the centrifuge. We are getting the 3 different layers. We are transferring that upper aqueous layer into different F&F tube. Then we are adding ice cold isopropanol into that F&F tube using the micropipette and storing under minus 40 degrees Celsius for 1 hour. After 1 hour, we are removing that from the deep freezer and centrifuging at 5000 rpm for 15 minutes. We are getting the white precipitate at the bottom. Now we are washing that with 1 ml of CTAP wash buffer and 3 ml of 100% ethanol. Then we are air drying it for 5 minutes to remove the ethanol. Finally, we are adding 50 ml of elution buffer into the FNF tube and storing it. Next step is gel casting. Now we are taking 10x TB buffer of 10 ml into the measuring cylinder. And adding 90 ml of distilled water for making 1x TB buffer. Then we are pouring that into the 250 ml conical flask. Now we are weighing 2 gram of agarose. Solving that 2 gram of agarose into the 100 ml of 1x TB buffer and mixing it thoroughly. Then we are keeping it under microwave oven for 1 minute to get crystal clear fluid. Now we are removing that from the microwave oven and keeping outside for cooling. Then we are adding 5 ml of ethidium bromide into the conical flask. Then we are mixing it thoroughly. Now we are pouring that 100 ml of 1x TB buffer into the gel casting tray by placing 2 wells in it. Then we are making 350 ml of 1x TB buffer for gel electrophoresis. Now we are removing the wells from the gel. Now we are placing the gel into the gel electrophoresis setup and we started loading it. In the first well, we loaded DNA ladder. Then in the remaining wells, we loaded the DNA samples. Then we have started running the gel electrophoresis under 100 volt. After 1 hour of running, we are removing the gel from the gel electrophoresis setup and moving it to the gel documentation. We have placed the gel into the gel documentation setup and on the UV light. Now we can clearly see the DNA bands present in the gel loaded. Finally, we are discarding the gel